All right. So tonight's event is titled, and we're going to talk about this title in a second. This is important, but what the title we gave it, the title that I gave it was Bats: Possible Cause of and Cure for COVID. And we have a very special guest with us tonight. Dr. DeeAnn Reeder is a professor of biology and animal behavior in the Animal Behavior Program at Bucknell University. While bats are among her specialties, her teaching interests include wildlife and emerging diseases, and among her research interests are disease ecology and white nose syndrome in bats, which I can't wait to hear more about. Dr. Reeder, welcome. Thanks for being our guest tonight.
Thanks very much. I really appreciate it, Mike. I'm really happy. I'm always happy to tell people about bats and about the work, and particularly how it relates to COVID.
Yeah. Let me just explain what I said about the title, because so I gave it the name, and I was drawn to the alliteration of cause, cure, and COVID. But what I sort of stepped on was blaming bats when I really shouldn't have. So why don't you jump onto that?
Oh, well, I'll talk about it for sure, right? The idea that that when an animal is a reservoir host, right, as many are, and you know, all animals have viruses. Bats have lots of viruses. Bats have coronaviruses. Bats may or may not have SARS-CoV-2, which is the virus that causes COVID-19. But bats have had them for thousands of years. Pandemics are on the rise. Outbreaks are on the rise because of human activity. So I'm going to address that a little bit with my slides. So I just had to give you just a little bit of a poke, I think, on the cause, because it's actually really prevalent. It's hugely prevalent. We see it all the time. And sort of from a conservation perspective, we're trying to be a little balanced. But I think I will just start and share my slides with you.
That'd be great.
Great. Okay, and let me present them. And it's in the wrong view again. There we go. All right, so I've retitled it Lessons from Bats. And I want to tell you, firstly, how amazing bats are. I'm going to move my camera over, so I'm going to look at people and not my slides there. I'm going to tell you about how amazing bats are, and then the links that we have to viruses, what we do know about them. So here, this is an epauletted fruit bat. It's one of my favorite bats in the world. It's one of my study species where I work in Africa. But I wanted to start being sort of a spokesperson for bats and how extraordinary they are. Today, the current total number of bat species recorded is 1,422. That actually is updated on a regular basis at batnames.org. It's a site run by a colleague of mine, Dr. Nancy Simmons, at the American Museum of Natural History in New York. She keeps track of new species and taxonomic revisions. About 1 out of every 5 mammals is a bat, and they're incredibly diverse. We find them all over the world, everywhere except Antarctica. Just really, really amazing. And they're really attuned to and adapted for very specific diets and very specific habitat types that they live in. For example, this is a fishing bat from Costa Rica. And as you can see, he has enormous feet. That's a unique adaptation for that. Bats have got lots of great things that they do, but this is the most important. So bats are the only flying mammal. So they're the only volant mammal, the only one that's ever evolved true flight. They did that about 55 million years ago, more or less. And it makes them ecologically unique, and it makes them physiologically unique. And it's both of those things together that I think tie them to some diseases that we tend to be really interested in. So just to give you some of the incredible diversity, most bats are insect eating, and as such, they play huge roles in balancing our ecosystems. We talk about ecosystem services sometimes, which is putting a dollar figure on what it is that a particular animal does for us. Bats save us millions of dollars in pest control. Huge amounts of money. So here's one. This guy's taking it a little bit to the extreme. This is a pallid bat from the American Southwest, and he's eating a centipede. There are bats that eat fruit, and as such are major seed distributors, particularly in the tropics. And they'll eat those seeds, eat the fruit, pass the seeds as they're flying over some forest gap, and really are responsible for helping regenerate forests. So key seed dispersers, also key pollinators. This is a lovely bat from Thailand, pollinating a wild banana species. So they play significant roles in that as well. Just incredible. And this diversity of feeding types and echolocation types, which we didn't even talk about, and differentiation by different habitat types has resulted in a variety of appearances. I love bats. I think they're amazing. Their reproductive patterns are crazy. Their color patterns are crazy. They just do amazing things. But I have to admit that, in fact, some bats are so ugly that maybe only their mothers love them. This is one of those examples. It's a ghost-faced bat from South America. Sorry, actually from the Antilles. This is a wrinkle-faced bat, really interestingly shaped face and skull, and we don't fully understand all of it. And here's a tube nose bat. So bats are incredible. They come in all shapes and sizes, and just really amazing. One of my most favorite things to do in the world is to hike into a forest and set a net, because for me, it's like Christmas. You never quite know what you're going to find, and it's just a delight to have them in your hand. This is a net that I have set across a small pond that I found. I hiked about 6 hours looking for water, and I found some. This is in South Sudan at the border with Congo. So one of the areas that I work. We also will set it deep in forest. So this is what we call a triple high net system. It goes way up high into the sky, and we can catch the bats that are up in the canopy, as well as those flying down at the bottom. And we just, you find amazing things. You find things like this butterfly bat, really beautiful, sort of resembles a monarch butterfly. You find things, we found this species and put it in a new genus. We recognized that it was really quite unique. This is my colleague, Dr. Babakir from Sudan. We called it a badger bat. You can Google badger bat. The media decided they wanted to call it a panda bat. So if you Google panda bat, you'll find all kinds of crazy stories about this animal that we found in South Sudan. So bats are just amazing, and I love working with them. This epitomizes how I feel about bats, and I think how rodents feel about bats. But I really put this slide up there to illustrate the point that bats are in fact not flying rodents. So rodents have a lifestyle that is fast, and they die young. And so they'll have litters multiple times a year. They'll have multiple offspring in each of those litters, and then they die when they're 2 or 3 years old. Bats do the complete opposite. Bats can live up to 30. Some are documented even older than that. And they typically have 1 pup a year. Here's that lovely epauletted fruit bat that you saw at the beginning. She has this 1 pup on there. The pups at birth can weigh about 1/3 of mom's weight, right? Which is like me giving birth to a 40-pound baby. And then they will attach to the teat and then hold on with their back feet, and mom will fly with them all night long. Takes an incredible amount of energy to raise these pups. And bats will spend, whether they're eating fruit or insects or whatever, they will spend the majority of every night out searching for food, and can eat an incredible amount of food. It's this flight feature that we think may play a role in their ability to serve as reservoir hosts. Now, it's a huge debate scientifically as to whether or not bats really have any more viruses, or any more likely to have viruses, than any other animal species. All animals have viruses. And so it's really a sort of a battle going on in the literature right now. And but what does appear to be clear is that bats appear to be reservoir hosts for viruses that we are especially interested in, viruses that have significant zoonotic potential, right? So zoonotic viruses are those that emerge from animals and spill over to people. The reservoir host is the animal in which that pathogen lives in nature. It has very likely co-evolved with that pathogen for thousands and thousands of years. And they have a nice relationship, and they have come to a stable relationship. And by and large, the reservoir hosts don't get sick from that pathogen. But bats do appear to be reservoir hosts for pathogens of what we call pandemic potential. In this case, this fruit bat that I work with is we're working with it in the context of the filoviruses of Ebola and Marburg virus. This picture you probably recognize from the news. This came out of the big West African outbreak that we had around 2015. Really, really horrendous pandemic. Even made it to the United States, but 90% mortality in people. But we were able to tamp it down. There've actually been 2 additional Ebola outbreaks since then, significant ones, but we don't hear about them very much in the news because they didn't leave Congo. But in any event, so I'm interested in these bats from the perspective of wanting to understand how can you be a reservoir host for these pathogens? What happened in the evolutionary history with bats and their viruses that somehow allow them to host the really nasty ones, the big things. And it's really all about flight, right? Remember I said that bats are ecologically unique, bats are physiologically unique. The fact that they fly all night long and it does things to them physiologically. One of the things that it actually does is generate a fever. So we call it the flight is fever hypothesis. My colleagues Tom O'Shea and others came up with this idea in 2014. And when it first came out, we all went, really? I don't know. And then a lot of us have sort of bought into it. The idea is that every night bats are having a fever for hours on end. They might take their body temperature up to 102, 103 and stay that way for hours. And the idea is then that the pathogens that have coevolved with them have been able to have their own genetic modifications, right? So it's always this sort of little coevolutionary war. Pathogen gets a leg up, the animal figures out how to fight it off. And then the pathogen has some other mutation that allows it to better infect cells. And then the animal figures out how to fight that off, right? Most pathogens, when they spill over to humans, right? What's the first thing we do? We generate a fever, right? It's really good. It actually helps stop viral replication, helps stop bacterial replication, things like that. A fever is a really adaptive response. But say you are Marburg virus or the first SARS virus or one of these ones that we know the bats have, you have evolved in an environment in which that animal is already having a fever every night. So you have had some mutations that have allowed you to persist at that temperature. So you spill over and are now in a human host as a new secondary host, and the human generates a fever and you say, I'm okay with that, right? So the pathogens have evolved the ability to persist at these higher temperatures, we think, because of bat flight. And there's a whole bunch of other things that go along with this. There's some really interesting immune modifications that we think bats are doing that are also a part of this this story. And this is where I want to do a little bit of my soapbox and talk about the fact that yes, bats may in fact harbor many pathogens. Some of those may be things that spill over to people, but bats have had these for thousands of years, and spillover and epidemics and pandemics are on the rise. So what's the difference there? The difference is human behavior. So the likelihood of a pathogen spilling over from a bat to a person increases the more contact you have between bats and people, and the more habitat disturbance you produce. This is a picture of Pteropus vampirus. It's a giant flying fox, and these are all, of course, dead animals for sale in a market. And so I just want to kind of run down a list of some of the things that people do and how those play into increasing spillover risk. One of those are the food choices that we make. You saw the animals there in the last photo, right? Anytime we eat a wild species, we call it bushmeat. Not all bushmeat is bad. I like venison just fine. That's bushmeat, right? But it's hunted sustainably, and it's been, hopefully butchered in ways that aren't spilling over pathogens and so on, right? So not all bushmeat is bad. Not all bushmeat is poaching. It's just the term that we use for if you're eating wild animals. One of the things that we see across China and Southeast Asia is the presence of live animal markets. And these we think played a huge role in the original SARS outbreak in 2002, 2003, and then potentially in the COVID-19 spillover event. When you have these live animal markets, sometimes called wet markets, they exist because there's a preference for very, very fresh meat. And so you buy an animal when it's still alive and it's butchered right there on the fly. But what happens is you have cages and cages and cages of animals piled on top of each other, and you might have a dog and a pig and a chicken and a bat and a civet cat and a snake, and they're all mixing body fluids. So it's really, and then somebody comes through with a hose and sprays everything, and everything gets aerosolized. So they're really, these bushmeat markets, particularly when they're live animal markets, are significant spillover risk. Sometimes we see wildlife being used in traditional medicine. There's risks there for that contact with wildlife. Ecotourism can be a problem, particularly for bats, because of cave and mine visitation. Global travel, also another issue, right? So it used to be that, you know, you would have an Ebola outbreak in a particular place. The village, nearly everyone died, but nobody was leaving, right? Nobody went and got on a plane to Toronto. And so our global travel is dramatically increasing the likelihood of spillovers and then epidemics and pandemics. Exotic pet ownership is an issue. Encroaching on natural habitat is a huge, huge issue. There's plenty of animals, including some of these big fruit bats, that when you destroy their natural habitat and move your village in, they're perfectly happy to stay. And there's actually some evidence that suggests that the ones that are staying close to people are not as healthy. And so that also can generate some spillover things. We know that, this is really well documented in the Lyme disease system in the American Northeast. We know that when we lose biodiversity, the increase of Lyme disease spilling over, getting into people increases. So that generates it as well. And then climate change is wreaking havoc on disease ecology patterns all around the world. What do I mean by spillover? And this is the thing that we often don't really understand. And we have practically no information for COVID-19 about how this might have happened. You have to have some way in which an animal is in contact with a person. So this is the hypothetical transmission pathway. Parts of this we don't know for Ebola. Shows bats and chimps over here on the left, and a little thing called a duiker, which is a little antelope, and then a gorilla over here. So we think there's this, we call this the sylvatic cycle, maybe moving from a reservoir host, which has actually not been proven to be bats. Most likely it's bats, but we're missing some of the final data there. But in any event, you have a hunting incident and that comes home. And you might have a variety of cultural practices that then lead to spillover one to the next, and so on. But for most pathogens, we just don't know this. For COVID-19, we understand what human behaviors are driving the spread one to the next, we think, but this whole piece over here, we don't really yet know, nor do we fully know what it means to be a survivor and what the long-term consequences for that might be. We have some ideas about what might be happening with COVID-19 because it's not the first coronavirus to spill over into people. If you're around in 2002 and 2003, you probably remember this outbreak. It was called sudden acute respiratory syndrome. And it was a global, it was a pandemic. I mean, it made it to Hong Kong quickly. It made it, it emerged out of southern China, made it to Toronto really quickly. It made it all around the world and caused a sudden acute respiratory syndrome. Everyone assumed it was a new flu. But it turns out it wasn't, and then they discovered that it was a coronavirus. Has this very telltale anatomy of the virus that you can see under a microscope, but it wasn't even the first. Well, what we mostly knew about coronaviruses before SARS is that there was this one called 229E that causes the common cold, but there's other ones that may have spilled over, you know, hundreds or decades or at variable times. Right? What you should be noticing here is that in many of these cases, we actually think there may be another intermediate host that is required. And that's why we have all these question marks down here. Is the reservoir host really a bat? Probably. But did it potentially get into some other animal? I think this is supposed to be a drawing of a pangolin. It's not a very good one. Was that somehow involved? I think probably not, but we don't really know that yet. And what are these pieces? And in order to prevent the next spillover of SARS-related coronaviruses, we have to figure this piece out. There are dozens of SARS-related coronaviruses in bats in southern China. Dozens. There we go. This article came out a few years ago, came out in 2017. It took us that long, from 2003 to 2017, to find the sequences in bats that allowed us to say the original SARS came from bats, and they were able to identify that. So that was a long time. It may take us a very long time to figure out exactly where the SARS-CoV-2, right, that very closely related, where it came from. But people have been saying since right after the original SARS outbreak, when we were discovering throughout southern China multiple different types of SARS-related coronaviruses, ticking time bomb has been a constant thing in the literature. You know, it's a variety of scientific papers in which the call was being put out of the risk of this. And, you know, people saying things like it was only a matter of time, and you know, people didn't listen, or at least policymakers didn't listen. And then we had it. This is from a few days after the outbreak started. If you look up in here, so this is a family tree for viruses. We call it a phylogenetic tree. Up here, this is the original SARS virus, one from a civet, and the rest of these are human cases. Fully sits within the same branches as all of these others. We call them SARS-like coronaviruses. These are all been found in bats. And so it sits in that same family tree. And then a little bit farther out, not this sort of maybe distorts it a little bit, still very closely related to the original strain. This is our new one, SARS-CoV-2, and these were the first cases in Wuhan. And then here is a bat sequence. The bat's about a 96% match genetically, which is sounds close, but not nearly as close as one would need to have a smoking gun here. Here's this other interesting thing. Sorry, here's our COVID sign. Here's our pangolin. You know, I think that before COVID-19, people didn't really know what pangolins were, but I think they know now, right? I always call them armadillos on steroids. They're an extraordinary lineage of armored mammals. The problem here is that these pangolin sequences that were found, these actually weren't even found from that same market. These were found in the illegal wildlife trade, confiscated animals. And so we just don't know. Were they in the proximity? Is this just another thing that it's going to take us quite some time to figure out? So into this milieu, so in the middle of COVID, right? So we have bats and we have COVID that's emerged, and it's really significantly tied to bats. I study bat immunity in relation to things like Ebola virus, and white nose syndrome. And a colleague of mine called me and said the National Science Foundation really wants to fund studies on immune responses in bats. And so I said, okay. And I called the NSF and I talked to my program director, and then I talked to my colleague, Ken Field, also at Bucknell. He's a card-carrying immunologist. You can see he's usually with the pipetter, and I'm usually the one out in the field. And I said, well, you know, what samples do we have? Because we can't go to China right now. We can't go anywhere right now. And so what we did was we went freezer diving. Whenever we euthanize, if we have to euthanize animals for studies, we save every tissue in every possible way. And we share all those tissues really widely with the scientific community. And so we decided that from the fruit bat that we have and from our little brown bat, some studies from Pennsylvania, that we had animals that have been preserved in tissues that would allow us to study coronaviruses. And so what we really wanted to understand is how do bats control coronavirus infections? And my running joke is always, if you want to know how to survive a coronavirus infection, you want to ask a bat, because they know how to do it. And they do it largely through something we call immune tolerance. So they have developed this relationship. They'll tolerate it at low levels. It doesn't kill them. They don't kill it. And it works out just fine. And they never immunologically freak out. They don't have what we call immunopathology. Here's the two species. You saw this handsome beastie already. This is the epauletted fruit bat. This actually comes out of our work in Uganda. And here's our little brown bat that we have here in Pennsylvania. And we already know about all the coronaviruses that they have, but we're going to go find more because we're going to do deep sequencing and figure out what are all the coronaviruses that these bats have and get their full sequence. And then we'll look at the bat immune response to them. This is a technique. Let me try to just put this a little bit into layman's terms, we're going to use a mechanism that allows us to read the entire code, genetic code, as it is in their RNA molecules for both the pathogen. Coronaviruses don't have a DNA genome like we do. They have a genome made of RNA. And then, so we can look for coronavirus RNA and figure out the sequences. But in the same exact samples, we can look at the bats' gene expression. So if you remember central dogma, you go from DNA to RNA to proteins. We're looking at the RNA. We're looking at what genes are being turned on in bats in relation to the particular type and level of coronavirus infection that they have. So it's just a technique that's going to give us all the letters. It's going to spell out all the letters and all the RNA sequences that we find in the bat's tissue. Two goals that we have there. One is to discover more coronaviruses from them, to fully quantify what are all the coronaviruses that these species carry. And then the second is to say, what are all of the genes that are being expressed? And in particular, we are interested in the immune genes. So the idea is, and I'm sure you've heard some of this in the press and in various reports, what is happening in the human cases with COVID is that for the people who go on to develop severe infections, their immune response to the pathogen has been really quite different. And it happens at about day 5 or 6 or so. And this reporter called it immune systems gone haywire. I call it an overly exuberant immune response. Sometimes we call it immunopathology. So the idea is that you want to mount an immune response that is sufficient to knock out your pathogen, but not so strong as to harm yourself. And that's what appears to be happening here, is that we move, it's this delicate balance between, for example, getting inflammation started, which helps us fight infections, but not having too much inflammation. So you have to sort of put the brakes on it. And that's what bats do. They know how to put the brakes on. So they know how to generate a sufficient response to the pathogen, and then keep those brakes on the system. It's sort of like holding your gas pedal and your brake pedal at the same exact time immunologically. And the goal is then that what we hope to find is the mechanisms that allow them to not develop immune pathology. What is it that allows that? What keeps them from having these overly exuberant responses, like the people who are dying of COVID? And can we somehow, in uncovering the immunological mechanisms, then make suggestions for what therapeutic targets might be, for example. And so that's where we are. That was probably a little bit longer than I had intended, but I am now very happy to start the conversation, and I will stop sharing.
That's great. Thank you. I appreciate that. So people start putting your questions in chat, and we will get to them as we can. Here we go. Let me just back up a second, though. I'm trying to get the chat to behave itself and be on the right side as opposed to taking up the whole screen. Okay. When I read about COVID and where it may have jumped, it seems to be taken as conventional wisdom that it came from bats. And recently I read the New York Times, they said horseshoe bats, as if it was known. Or I guess they said likely. So you seem to be more questioning about it. I guess they haven't found this particular virus in any bat yet.
They haven't, and we might not ever find this particular virus. It's a bit of a needle in a haystack. Right. So horseshoe bats, they're bats in the genus Rhinolophus. They have a little kind of a thing on their nose. That's why we call them a horseshoe bat. And it's a particular lineage from the family Rhinolophidae. It is where, and with a handful of other bats that have also been found in, but it is where the SARS-like coronaviruses live. So I don't doubt that bats are a part of the COVID-19 story. But whether or not this was a pathogen that spilled over in a bat 50 years ago into some other wildlife host, and right. Then has now mutated in that second host and now spilled over to people, right? That's a piece of what we need to figure out. I don't think that there's anyone who studies bats and viruses who doesn't think that rhino-lophid bats won't play a major role in this story. But the data, that the closest virus that we found, being a 96% match for SARS-CoV-2, the human virus, suggests that it's been about 50 years since that bat virus and that human virus shared a common ancestor.
Oh, wow.
There's a lot to be sorted out still.
Right. And your description of wet markets really helped me a lot, because I was always curious about the name. But
Yeah,
the way you described it was helpful.
Yeah, people say different things for it, for why it's called a wet market. Generally, we talk about wet goods and dry goods, right? So dry goods is like your bags of rice and cereal and things like that. And wet goods is everything else. That's, I think, where the name originates, but people also use it to describe other things about the market. Really, it's really quite challenging. And honestly, I think there are constant spillovers waiting to happen whenever you have those live animal markets, particularly if you have animals of multiple different species all in the same area.
And another interesting thing I want to come back to also is on the possible, looking to bats as possible insights into how we get through the pandemic and how we treat COVID, is that it seems that children have a more bat-like physiology. So children maybe run hotter or differently. Can you help us work that out? So that, and this in part explains why it's not that children are immune from COVID, but that children are less impacted by COVID than adults are, especially older adults.
Yeah, so I am not a human physician or human physiologist either. But in general, young mammals regulate their immune systems more effectively than older mammals do. And so I think that they thought from the get-go that they weren't surprised to see that children were suffering at much lower rates and were more likely to be asymptomatic. But there are other pathogens that preferentially can kill children. And for example, children under 5 tend to be especially vulnerable to pathogens like malaria, for example. So it's not it's not ubiquitous. It isn't going to happen in every system, but it surely seems to be something that's happening here. And I think that in general, they are, what I presume is happening is that they're also not doing that immunopathology. They're just not triggering those other immune pathways, which probably means that they're dropping the viral load much more quickly. But I think, there's so much that remains to be seen.
Right. And you received a rapid grant from the National Science Foundation. So how rapid is a rapid grant? Like when do they want to?
We were completely blown away. I mean, so I think that in May, we first talked to our program officer, and she said, by rapid, I really mean rapid. And we're like, okay, well, what does that mean? Because normally you send off a grant and 6 months later you get a score, and 6 months after that, you might get some money.
Right. And
with the federal government. But rapid grants, we submitted it within 10 days, we had a decision, and then within like 6 days, we had the money.
Wow.
Or the grant signed to the university. And then so we hired somebody to do a lot of the lab components, and boy, we got rolling. I've never seen anything move so fast.
And how far along are you?
So we are, we've identified all of the samples that we want to use for the study. It's all gut tissue. So for most animals, coronaviruses are infections of the gut. Respiratory is unusual. It's something that the SARS-like coronaviruses tend to really like respiratory tissue because of the receptors they bind to that are prevalent in the lung tissue. But so for us, searching for other bat coronaviruses, it's the gut tissue. So we identified those and did our freezer diving. We got our last shipment of samples in from Uganda for the fruit bat work. And we are doing at the moment what we call optimizing the RNA isolation. So we literally, it's, grinding up the tissue. And well, the first thing we have to do is pathogen inactivate the samples. So potentially our African samples could have things like filoviruses, Ebola in them. So we have to pathogen inactivate them and then go through a whole series of chemical steps, some of which is literally grinding up tissue to pull out all of the RNA. And then we have to decide if we think that RNA is good quality. And then we actually send it out for the deep sequencing. And then once we get those out, probably in about 6 weeks, we'll have billions of data points, lots of letters. And then we do the bioinformatics component of it, which is to actually go in and try to understand what viruses are there and what the bats are doing immunologically.
Wow. Okay.
But it's supposed to be done in a year.
In a year. Okay. Got it. Good. Oh, good. Well, good luck with you.
Rapid. Yeah. Yeah.
I'm going to start going to some of the questions in here and sprinkling in my own, but Nicole wants to know, to have a time frame idea, how long would it take roughly to create immune tolerance if in fact we could eventually have that? I imagine it took bats many years to develop it.
Yeah, so it's really,
decades, centuries. Anyway, go
yeah,
ahead.
so it's often not, I mean, immune tolerance means to not overly respond to a pathogen, and particularly to not have immunopathology. So any individual could have what we call a tolerant phenotype, where they're just not going to over respond to it. And they may, in fact, not even be trying to fully eliminate it. So any individual could display a tolerant phenotype, but if you're talking about bats in general, with their pathogens in general, we're talking about thousands of years of coevolutionary time.
Evolution works
you know, when we
slowly.
think about, you know, pathogens and hosts, it really is this coevolutionary war of, you know, you have a pathogen has a mutation and it's replicating at a much faster rate than we are. And then we have to have, you know, we also have to have some sort of mutation that allows us to then fight that. And you go back and forth, and you end up at a stalemate. And that's really what immune tolerance is sometimes described. So sometimes people can have a tolerance response to a particular pathogen, but in general, immune tolerance to viruses in general is something that's evolved over hundreds of thousands. And
the Rainer family wonders, how exactly does a lack of biodiversity connect to spillover?
So this is sometimes a little contentious because it's not been shown in every system. But what you see, so look, for example, at a pristine patch of habitat somewhere in New England that has deer and Virginia opossum and chipmunks and a couple of bird species and a little deer mouse on it. And that's its normal biodiversity that it would see. It's an undisturbed habitat. And it's an interaction with the ticks that are carrying the bacteria that causes Lyme disease. And when you have all of those species in that environment, some of those species, particularly the opossums, are really good at grooming off the ticks, and they keep the levels of Lyme disease down for all the other animals in the forest. When you start removing some of those species, so you start disturbing the habitat, and some of the bigger stuff just goes. Right, and what turns out to happen, so if you're, say, for example, in this instance, if your Virginia possum was the one that was controlling the infection levels for everybody and it's gone, so you've lost some of your biodiversity, we now see Lyme disease flare up in all the ticks that are there, and we see then increased cases of transmission to humans. And it's been shown in some other systems where once you start losing species, it's sort of like it's called the dilution hypothesis, this idea that all of the species being present there are sort of diluting or buffering the pathogens being able to sort of move between them in a way. And once you start disturbing that delicate balance that's evolved over, you know, millennia,
right,
then you change the disease cycling. But it's a little bit controversial. In some systems, it's very clearly been shown, in others, not.
Right.
Good question.
Right. Interesting. Yeah. I guess the habitat loss is easier to understand how that would cause spillover, because we're pushing into, you know, deep forests where people never were for a very long time, and we're bumping into new populations of animals that we've not come across before.
Absolutely. I mean, you look at a Nipah virus that happened in Malaysia. You had people cut down forests and move in pig farms, and the giant fruit bats that lived there decided that they still lived there. And so now all of a sudden you have these giant fruit bats interacting with pigs and dropping their spats. So fruit bats will chew fruit and then spit out the seeds and the pulp and all the things that they don't want. And then
Which
a
is
pig
full of viruses, probably. Right.
Right. So they're shedding in their saliva, and then a pig would come along and eat that. And plus the bats are flying over and they're peeing and they're pooping into the pig feed. Right. And so so in that instance, all of a sudden here's this virus, Nipah virus, that's been in these bats for thousands of years, and all of a sudden it has a new host. It has a pig. Turns out pigs are really susceptible, and they become sort of virus production farms. You know, I mean, they're making this, and they're frothing and they're barking, and then all the pig farmers get sick. And that connection, until you all of a sudden cut down the forest, move the pigs in, so now you have a new intermediate host, and this virus happens to go here to here, even though it couldn't go here to people without that host.
Right.
Right. So habitat encroachment is, I think, the leading thing that's pushing the increase.
Yes. So environmental people like me have been talking about deforestation for decades, of course, and never because we want to stop pandemics. That was never one of the
It's just another interesting.
All right. I may have stepped on Sarah Whitman's question as she was probably typing it as I was asking you. I read that bats' immune system is more similar to human children's immune system. The bats are able to not get sick from coronaviruses, like children seem to be much less susceptible from getting sick. Can you talk about that? So I think we kind of did, but if there's any PS or...
Well, I think it would just be really interesting. We're actually now, there have been a couple of papers published that have
Yeah.
really looked in depth at what adults are doing in the response. And I assume that there are medical professionals who are doing clinical studies in children and actually looking at what's happening in their immune response as well.
Yeah.
And so I'm hopeful that we'll actually be able to eventually get to those answers. It would be really interesting if children are using some of the same mechanisms to not over-respond.
Mm-hmm. And by the way, the piece that I read, and it was a New York Times Sunday magazine I got next to me, it's called The Cytokine Mystery. So if you Google cytokine mystery, bats, New York Times, you'll get this article about, and it'll talk about kids and bats.
Cytokines are the chemical messages of the immune system. And part of this overly exuberant immunopathological response is what we call a cytokine storm. And you sort of, it just sort of snowballs. And then all of a sudden the levels of cytokines are extraordinarily high and they trigger a whole series of bad immune outcomes.
Mm-hmm. Kim Eberly Wang says the human immune system is very complex with many cell types. Is a bat's immune system similar in terms of the types of cells and their functions, B cells, T cells, et cetera, to a human immune system? Thank you, Kim. Go ahead.
That's a great question. It largely is, although in some cases we don't know because we don't have the markers for bat T cell subsets. So I can get Bs and Ts if I'm lucky, but I can't identify helper T cells and killer T cells and all of those things. It's actually really challenging. We don't have the same tools that we do for humans, but we do believe that they have the same basic cell types. What has been interesting is that there are some immune genes that are missing in bats entirely that we think, actually there's one called STING that is responsible for dealing with DNA viruses. And then there's other cases where we have found that bats are turning some genes on at different levels and maybe sort of hypervigilant for viruses at all times. So it really is in that detailed regulation of all the different immune genes that we think that the biggest differences are being seen.
Mike, thanks you for the great presentation. My question is slightly tangential. Do you have any advice for setting up a bat house in an urban Philadelphia backyard? Is it possible / advisable? Is there a minimum requirement for open space or distance from structures?
Yeah, I love bat houses, but they don't often get occupied. So we put a lot of effort into them. And there are instructions. You can go to Bat Conservation International. They have a great site that talks about placement of bat boxes. It's also a company in Carlisle and PA called Bat Management that builds them and will actually come install them. And depending upon where you are, you want to paint them. If you're up in the north, we paint them black. Down in the south, we leave them natural wood because you're trying to create a particular temperature environment for the bats. The problem with bat boxes is that unless bats are looking for a new home, they're not typically going to occupy them. They are very happy to be in our attics and our barns, and some of them just to stay in trees. And so, yes, it's always great to put them up, but I think bats in Pennsylvania, especially right now with the numbers that we've lost from white nose syndrome, they're not desperate for roosts. They have plenty of roosts. So, I mean, I see people all the time. It's really common, like Eagle Scout Project. And sometimes they get occupied and they're great, little population centers of bats, but mostly I see people put them up and wait in vain. And you get a lot of wasps.
So if people are interested, they should go to Bat Conservation International.
Yeah, absolutely. It's a great site for information.
Yeah, COVID's all over the front page of their website too, if you want to dive into this a little more deep. And I think some of your photographs come from them. Is that where some of your photos come from?
Some of them, yes. So they're the man who established that Merlin Tuttle, sort of the Batman of science. And he—
I recognize the name, yeah.
Extraordinary photographer.
Yeah,
extraordinary photographer. And so he sells the rights to his images to scientists very cheaply.
And
so there's
a few people, only a few of us heard this as we were talking about bat houses before we actually started. And you would talk about the number of bats if you had a successful house, the number of bats who could be in there, which blew me away.
It depends on the size of the house, but if you put a big house up, you could have 1,000 bats, 2,000 bats, depends on how big it is. Most of the little ones that we might put on the sides of our house could easily get a few hundred.
How many of us would have guessed that? I would have thought 2 or 3.
Amazing. And all you have to do is you get a good occupied one, and then you can just sit there and do a count at night, have a little clicker, and you'll just be blown away at the number. And they'll just pack in.
But that also explains why diseases spread so quickly, because they're so close. Which is our next question from Joanna, to please elaborate a little on white nose syndrome. Is it bacterial, viral, fungal? Is it a global problem or only bats in the U.S.?
It is fungal, and it's a more complex answer for the where is it thing. So it is an emerging fungal pathogen of bats in North America. So the fungi's name is Pseudogymnoascus destructans. We call it PD. First arrived in the US.
Destructans is the species name?
Named it. My colleagues named it for what it does to the bat's wing. Yeah. First cases, we believe now are documented in 2007. What we initially first saw was bats in hibernation having fungal spores, it's like little mushrooms, right, growing out of their nose. And so we called it white nose syndrome. And then it started to spread, and it has spread, and it has spread. And you can actually go to whitenosesyndrome.org. It's a site hosted by the U.S. Fish and Wildlife Service that keeps up with the spread. It has extraordinary virulence. For some of our bat species in Pennsylvania, our early estimates for the little brown bat, which was once our most common species and is now listed as endangered, we've had about 90% mortality in the state of Pennsylvania from this fungal pathogen. It only grows in the cold, so it kills the bats when they're hibernating. Hibernating bats typically turn off their immune systems, because most pathogens also go cold with them. They don't need their immune systems, and immune systems cost a lot of energy to run. So the fungus sort of has found their Achilles heel, and it grows on them when they're hibernating, and it causes them to warm up, to come out of hibernation over and over and over again, and they burn up all their stored body fat.
It's deadly. Yeah, that's so sad.
But the good news is, so it has spread across the country. We have about 25 species of hibernating bats in North America. And it has spread widely across the country. Some of the species turn out not to be susceptible. Our big brown bat, another very common species, is doing just fine, as are some of the others, including the Virginia big-eared bat, which was really critically low population numbers. Turns out is totally fine, which is a godsend. Where it came from is the interesting story, and what we eventually uncovered, and this was with lots of European colleagues who said, oh, we've seen that. We've seen that forever. We always see that in our hibernating bats. Turns out that it likely came from Europe, and then there are also some Asian strains that have now been described, and the bats have probably had it in Europe for thousands and thousands and thousands of years, and they already had their own bottleneck a long time ago, and their bats are now tolerant of the pathogen. Mm. And the nice piece of news on white nose syndrome is that in the Northeast, the bats that we have in our remnant populations, what's left, appear to have some tolerance. So we got down to about 10% of our population, but they're persisting. And we hope that they'll continue to do well, but it will take decades, if not centuries, to bring
because they only have one baby a year. Or
one baby a year, and that pup has about, in the hibernating species, that pup has about a 50% chance of surviving its first year. Yeah. So it's slow reproductive rate. That's a great question.
But once they survive, then they last for decades, which is news. That was news to me. Great.
Okay. That's great.
Kim asks, also a scientist, is his household also a scientist in this household perhaps is working on a medicine to treat cytokine storm in humans? Are you able to test any potential coronavirus medications in bats?
Well, we have done, actually, in the context of our white nose syndrome work, we've actually done some drug trials to look for safety profiles, and actually we would have the fungus, and we did some experimental infections. For SARS-CoV-2, I'm not set up biosafety-wise to do any to do the experimental infections. They are actually happening in this country in a couple of labs. Some North American species are being tested to see if they'll get it. But sort of safety trials it would be interesting to see how the bat's immune response might work. But I don't currently have any bats on campus. I actually do have at Bucknell 3 flight cages and a whole series of environmental chambers that we hibernate our bats in. But at the moment, we don't have a bat colony But we have done that sort of experimental work before.
So just watching the clock, it's almost 8 o'clock. We're going to go past 8 and we'll go to 8:10, 8:15, or as long as you want to hang in there. We've got a lot more questions coming in. So we'll go past a little bit if you don't mind, everybody. Sarah, as going back to your comment about bats having a fever when flying every night, is their baseline temperature higher than 98?
No, well, that's the thing is they're really heterothermic. So what is their baseline temperature? You know, most mammals are what we call endothermic. They can generate their own body heat, and then homeothermic. So they maintain a constant body temperature, right? So we have our 98.5. That's where we are when we're homeothermic. Bats are endothermic. They can generate their own body heat, but sometimes they choose not to stay at the same temperature. And so they can get a fever sometimes, and other times they can completely drop it down. These fruit bats that I study in Africa, they'll go from 41°C, which is 102, I think, Fahrenheit, down to about 20°C, which is 55, something. I'd have to actually, you know, in real life, I work in Fahrenheit, and in science, I work in Celsius, and I don't move between them very well. So bats are actually constantly adjusting their body temperature. So it's kind of like, what's the regular body temperature? What's euthermic for a bat? Differs by species. We sometimes study what we call the thermal neutral zone, which is like, what's that little range of temperatures that they seem to be most comfortable in? But yeah, good question. But actually, this whole idea of, you know, what's happening during that bat fever immunologically is something that I'm hoping to test soon. I have a big proposal into the National Institutes of Health, who's funded some of my fruit bat work to actually study the role of fever.
So our question also is, does the higher temperature kill some of the virus load? Is that what happens?
You know, we just don't know. But tell the NIH to fund me, and I can answer that question in 3 years.
Right. Amanda put the Bat Conservation International Bat Box site up on the in the chat. Thank you, Amanda. Is the nighttime flight really a fever, or are they just warm from flying? I think we just.
Oh, actually, so that's a really interesting question. They are warm from flying. They cannot dissipate their body. They generate so much heat. That metabolic activity of flapping your wings all night long generates so much heat. You know, when you're running, you get really hot and you're sweating, right? So for them, they're generating so much heat, and they can't cool down fast enough because they keep, it's like, you know, they keep running. And they're, and so they allow themselves to basically just overheat. It's not like when we generate a fever, we're doing that in response to a pathogen, and we actually have a particular cytokine called IL-1. It's also sometimes been called a endogenous pyrogen. So it's the thing that tells your brain, you know what, you're cold. You don't think you're cold, but you're cold, and so you need to raise your temperature. It changes your whole physiology so that you get the chills and you actually generate more heat. So you're right. Maybe fever is the wrong thing to call it. It's just what they do. They can't cool down, and so it's just it's an artifact of the fact that they fly all night.
So it's not really an immunological response to them.
I don't think it's an immunological response to it, but what I think it's done is selected for pathogens that can persist at that body temperature.
Got it.
And we also do think that it actually has consequences for DNA repair and immune regulation.
Right. I had a whole page of questions in case our people were shy, and they're not, so I don't need my page of questions. But one of them I had was how you've developed your interest in bats. Eliza wants to know too.
Okay. So let's see. So I have, from the time I was, you know, I don't know, 4 or 5, I knew I wanted to work with wildlife. I would run home from school and watch Mutual of Omaha's.
Of course.
Just, you know, there were 3 channels, right? That's what you did. And
it was on Sunday night, right?
Yeah, I grew up traipsing around the Sierra Nevadas in California, from California originally, and I love nature and I love wildlife. When I went off to graduate school, I actually studied primates, and was interested in primate physiology in relation to behavior. And but they're really hard to study in the wild. And I am interested in animal physiology in relation to its ecology, and that's really hard to do in primates. And so I thought, you know, I was just finishing my PhD. I said, I need a different group of animals to work in. Here's the kinds of things I'm interested in, and what group of animals should I go work on next? And it turns out that bats were the answer to that. And I've always dabbled a little bit in immunology, but that's not my primary training. But the white nose work and then the African fruit bat disease ecology work sort of, you know, came together, and that's now what I mostly work on. But, you know, so I switched to bats 20 years ago when I finished my PhD, did a postdoc and then came to Bucknell as a faculty member, and I haven't left them. That said, I do work on some other groups. I do major conservation work in South Sudan. I have some chimpanzee funding to manage population there and track it, and I study the forest elephants there. So I do a handful of other mammals and conservation and biodiversity work in general. But when I'm wrangling an animal in the field, it's almost always a bat.
Joanna points out that 20 degrees Celsius is 68 Fahrenheit and 40 Celsius is 104.
Okay, all right. Well, that's what my bats are doing day to day. I'm going to write those down. You know, it's one of those things you never think to actually do the conversion until you have to say it to people, and then you're like, oh, I don't know what that is. Well, thank you.
James wants you to elaborate on the protection status of bats in the U.S.
Yeah, so that's actually been something that's been in flux because of white nose syndrome, right? So little brown bats used to be our most common species, and now they're endangered. Once something becomes endangered, right, then it gets regulated by the Endangered Species Act. And then that, you know, there's always politics that play a little bit of a role in that. And as each of the species that have been affected by white nose syndrome have gone through their own population declines, then there's been decision-making processes about establishing that. And once something is on that ESA list, then you can't just go, you know, cut down a forest or put in a, you know, a new line of electric poles or something without doing some impact survey work and tracking bats and seeing what's there and making sure that there's not a maternity colony of bats there or a hibernating colony anywhere nearby. So that's sort of the legal side of it. From a COVID perspective, what I haven't talked about is this notion of reverse zoonosis. So we have significant concerns of COVID going from people to animals, including wildlife. So I was on a panel of 12 anonymous scientists who helped the federal government through a decision-making process about the risks to North American bats, that bat biologists in handling those bats and then releasing them might potentially be giving bats COVID-19.
Oh, my word.
Yeah. And we came to the decision that it could be possible, and from a biological standpoint, in terms of thinking about the receptors that bats have and so on. But that we didn't think that it would be likely that the bats would then be able to give it one to the next, but we still felt that the risk was sufficient, that some work needed to be curtailed. And so actually there's a lot of field work not happening in the US and around the world. So if you have long-term studies, you can't put your hands on bats this summer.
That's so interesting.
So it's been a, you know, sort of a mixed blessing, but everybody decided that we had to use an overabundance of caution. My colleague who's running my work in Uganda right now, he's now in a full PPE to work with our fruit bats. And he has to explain to people, no, I'm not worried about what the bats are going to give me. I'm worried about giving COVID-19 to the bats. So what we don't want is new wildlife reservoir hosts being established, wherein that virus can now mutate in new ways.
Right.
We do know that it can go to cats, right? That transmission to house cats is possible, and there's some concern of it getting into feral cat populations in the US.
Oh my gosh. Okay.
It's a whole other scary thing.
It says that last week's talk about bees had the expert mention that honeybees are doing well, but many other insect populations are decreasing. Is decreasing insect populations affecting bats? Actually, they've called it like insect apocalypse. It's like,
yeah, they have. And actually, you know, there are people who study that, and I have to say it's not something that I keep my finger on. So I don't know whether or not people have suggested that. In general, bats are, especially in the US with our lower population numbers now, I don't think that we are seeing an issue there in terms of them getting enough food. But I don't know. I don't keep my eye on that literature. So it's possible. One of the other issues that happens, particularly in our insect-eating bats, is that they're exposed to really high levels of pesticides, because the pesticides are bioaccumulating in insects, and then the bats are eating the insects. So that's a part of that whole sort of insect apocalypse story. But whether or not they are not finding enough to eat, I don't know if anybody's, I'm sure people are tracking that and studying that. I just don't know.
We have time for one or two more questions. If anyone wants to put one more in chat. Beth wonders, are wind turbines detrimental to bats? As we talk about green energy and put turbines up on mountain ranges or hillsides. Yeah, go ahead.
Yeah, wind turbines are fantastic except for migratory animals.
Right.
Right? So we put them along ridge tops, right? Which is what animals are using to migrate. So you've got birds that are using it, and you've got bats that are using those. And it turns out that the species that don't appear to be as susceptible to white nose syndrome, because they're not packing and hibernating in caves, they're migrating, they're leaving in the winter instead of hunkering down in the cold. Those are the bats being hit by the wind turbines. So it is a significant concern. But it was funny. That was sort of the leading concern for bat conservation in this country before white nose hit, and then it's just sort of been overwhelmed by white nose syndrome. But again, if you look at my friends at Bat Conservation International's site, they'll have some good information there about wind turbines. Absolutely, we see mortality, and that's something that the wind turbine people are supposed to be tracking. Numbers of carcasses below. It's really awful. Most of the times the bats that die are not being actually hit by the blades, but it actually causes their lungs to implode from the air circulating around the blade.
Oh wow.
Yeah,
Okay.
it's not very pretty.
Let's end on a happier note. Let's go back to the badger bat. Now you helped name this species, right?
Well, so we found this in South Sudan, and it was absolutely a new bat to me.
And this is the panda-faced one for everybody.
Panda-faced one, really, really extraordinary, beautiful, beautiful animal. And I got back to the U.S. and talked to colleagues about it, and realized that it had actually been collected in Congo in 1937, and so it had been given a species name, but we decided that it was so unique that we didn't think it was as closely related to everything else in there, so we gave it a new genus name, and I gave it a name in the Azande language with the people that I work with there in South Sudan, and it's just been a great conservation story for South Sudan, which is always desperate for happy news. Because it's.
And what was the genus name?
Neumbaha.
Which means?
Which means it's rare or it's special.
Great. And what was the specific name? The species name was?
So the species name is Superba.
I remember, that's it.
So somebody had already actually named it Superba. They named it in Glauconectar superba, and we named it Neumbaha superba. I now have colleagues who disagree with me, so we're having a little debate in the literature. But
such a science.
Bat, great, yeah, a beautiful bat, really, really lovely story. Yeah, and there's nature is amazing.
Right. And I love when I see a scientific name for something that tells me something about the animal. So when you see Superb, I was like, oh, that's great. Yeah. That's a smart name. Yeah. Good. So good luck with your research.
Thank you.
And really appreciate your time with us. And we're all, I know everybody, we're going to be watching this story very carefully. Hopefully DeeAnn will come back to you at some point, maybe next spring when we see how you've done.
Okay.
But really thank you for your research, and thank you for your time tonight.
I really appreciate it, and I'm always happy to be an ambassador for bats.
You're getting lots of thank yous in the chat, so appreciate it. Thank you very much. Clearly, you must be a great professor. I just would love talking to you, so thank you so much. I wish I could take a class with you right now. I want to thank Amanda for being our Zoom co-pilot. I want to thank all of you, especially if you're a member. Thank you so much. If you're not, love for you to join. Come to additional programs. Next week is an art party here on Thursday night. We're open. Come for a walk. Come see if you can find little brown bats flying in our forest. If you do, please send me an email, mike@schuylkillcenter.org, because I've not seen one yet, and I'm desperate to see a bat. I've only seen like 5 monarchs, so there are a couple of animals I keep looking for as touchstones. I have not seen a bat yet this year, which is a little terrifying for me. So I want to thank you all again, and have a great night. And see, oh, by the way, one more thing. This is taped. So if you want to get a recording, we can get it to you. If you have friends who missed it, we can get them a recording as well. So thank you, everybody. And thank you again, DeeAnn. Thank you, Dr. Reeder.
Yep.
Have a great night. Bye-bye. Thank you, Amanda.